Well, thank you, Kelvin, and thank you, everyone. I'm really excited to be able to present here and really appreciate being asked by BioHive and BioUtah to be presenting. And so I think you'll quickly notice that the company, Known Medicine, is quite new. Giving a little bit of history about how we were founded, as Kelvin said, I do have a PhD in biomedical engineering. And while I was receiving my degree, I was incredibly focused on cancer tissue engineering. And so this was a really growing field when we think about what that really means. And so one half of that is going to be developing biomaterials. That's really what I was excited about, doing the chemical synthesis. The other half of that was cancer biology. How can these cancer cells from actual patients go into biomaterials in vitro and really replicate the body? And so this was something that I was asking every day and answering. And so as I was wrapping up my PhD, I started to think, what can I do with this technology? The data was incredibly compelling, but there was nobody really doing it in industry yet. I also was very familiar with a company that you'll hear about later, Recursion, and was understanding that high-throughput automation was incredibly powerful, as was data science, machine learning, AI, of course. And so I started to ask myself, can we translate these complex 3D models into something that was high-throughput and really replicative of the patient body? And so in early 2020, my co-founder, CTO Skelly, and I were talking about what could a company look like in which we merge these models with data science. And so Katie Rose has a background in computer science and biomedical informatics from Stanford and was a data scientist at Recursion at the time. And we realized that our skill sets married together would make the perfect company for identifying the best treatment for every cancer. Looking back, as I said, we have been around for just under 2 years, and so we're truly a COVID company. We connected in February of 2020. We then applied to Y Combinator, and that's an incubator based out of the Bay. And so we applied, we were accepted, and somewhere between there, I managed to defend my PhD. I was in North Carolina, and Katie Rose was in the Bay, and we came back together in Utah. And so Utah's been an incredibly important place for our company, and we're excited to continue growing here. After YC, we then closed our initial seed round. We employed our first scientist in early 2021, and then moved out of the Altitude Lab. And so Altitude Lab was an incredibly important place for us, and something we really saw as a huge benefit for being in Salt Lake. We knew that during Y Combinator, we needed to get our proof of concept up and running. And so how could we show that data science really can come together with these complex 3D, previously very artisanal models and have something powerful? And so Altitude Lab was there to offer us lab space and help us get off the ground. We then moved out of that lab space in early 2021. In the last year, we closed a second seed round and have been able to partner with many of our first clinic, as well as many others, to be able to measure patient-to-patient variability to further understand cancer. And I'll certainly be diving into our technology here in a minute. And then finally, in 2022, we've been able to expand our lab and office, and we're now 16 employees. I love these pictures. So the first is Katie Rose and I with our eBay purchased biosafety cabinet getting set up in Altitude Lab. And this was before Altitude Lab was even built out. And then those other two images show our team today working in data review and the automated lab system that we've built out in the research park. And so we're incredibly proud of how far we've come in just under 24 months. And building in Utah, as I said, has been incredibly important for us. Altitude Lab allowed us to get off the ground, get that initial seed money, and build the company that we are today. BioHive and BioUtah have also been incredibly important to us. And this is because we're not Utah natives. Coming from the East Coast and the West Coast, we were fully embraced by this community. I have the opportunity to sit at dinners with other CEOs, meet COOs and CTOs that can really drop quite a bit of knowledge on us, and have been able to find business strategy and the help that we need here to become a really, truly successful drug discovery and development company. And we're incredibly grateful for the people we've met and the community we continue to be a part of. And so Known Medicine is building a predictive engine to capture patient-to-patient variability and develop the best drug for every cancer. Today, unfortunately, it takes 10+ years and oftentimes billions of dollars to bring drugs to market. However, and this is an incredibly striking statistic to me, that from phase 1 to market, only 3.4% of oncology drugs will make it all the way through. And so we can discover these incredible drugs, but we can't put them in the right patients. And this is incredibly frustrating, not just for me as a scientist, but I'm sure for every single one of us who has watched a family member or friend go through clinical trials or drug treatments only to not respond. And so how can known medicine play a role in identifying the best patients for every drug, both discovered and developed? We're able to do so using our 3D cell culture models married with data science to measure that patient-to-patient variability. This really requires 2 pillars, the first being microtumor outcomes. So we're able to take patient tumor samples, break them down, and then make thousands of microtumors. These microtumors can then be tested, or drugs can then be tested on them, so we can understand how will this patient respond to many different drugs. Now, imagine if we have hundreds and then thousands of these patient samples in which we can understand patient-to-patient variability for every drug. And so we're able to take these functional outcomes generated from those microtumors and then combine them with biomarkers. And so I think we're all very familiar with biomarkers and the power they have. We all know about genetic sequencing, we've heard of proteomics, and something about immune cells likely as well. And so we're able to measure genomics, proteomics, and immune cell populations and marry those with the functional outcomes of microtumors to see which drug will you respond to and why. This is incredibly powerful when we're developing drugs, as we can run essentially a phase 2 or phase 3 clinical trial on our platform without a patient ever having to receive that drug. And so we can identify the best biomarker, likely many biomarkers, that that patient must have to then receive the treatment. And then as we move into discovery, with more and more patients that we run on our platform, with more and more novel compounds, we're able to understand what secondary or tertiary biomarkers we need to be discovering drugs and developing drugs around. And as I said, the platform is receiving patient tumors. Right now, we receive them from across the country. Most notably, we have a Duke University partnership that we've recently announced. And so we receive lung and colorectal cancer tumors and match blood samples into our lab. We then break down those tumors, get all of the cells out, and those are cancer cells, immune cells, stromal cells. We then put them all back and make thousands of microtumors. We then treat them with chemos, targeted therapies, and immunotherapies to ultimately understand how they'll respond. We're able to measure that response through image analysis. And this is something that's certainly complex when you understand that we're looking in 3 dimensions. And so it's not as simple as looking at how are these cells interacting in 2D, but now let's look at the 3D space. How are they interacting, changing, moving around, looking at phenotype in response to that drug treatment. Today, we've received over 165 tumors into our office and lab. And of these, what's really cool and incredibly important for development is that we're able to biobank these samples. And so biobanking allows us to then use these patient primary cells many times over in the future. And so as we have all of these biomarkers for each patient and these samples banked, we can continue to develop and discover drugs around these patient populations. Today, of all of those patients, we've been able to receive follow-up, and that includes what drug did that patient receive, as well as how did they respond to that drug in a subset of these patients. Of those, we've been able to correctly predict 5 out of 5. And so 2 of those patients were non-responders, and we were able to indicate that they would not respond to any treatment. 3 of those patients were responders, and that was a combination of both chemo and targeted therapy. We were able to predict correctly in all 3 of those patients which drug they would respond best to. And so although this is a small sample size, we're quickly increasing that and believe that we'll have over 1,000 tumors in the next year and a half. Our success to date is really exciting for being around just 18 to 20 months. And so the first is that we've been able to take an idea and build an automated high-throughput 3D microtumor platform. And not just create it, but generate functional outcomes in which we have validated. And that's cell lines, PDX, of course, but then also that clinical data. We've also expanded well beyond chemo and targeted therapies and are able to now use immunotherapies in which we're maintaining immune cells for over 5 days in culture, as well as being able to apply immunotherapies and observe changes in cell behavior. This wouldn't be possible, and the platform wouldn't be powerful without measuring patient-to-patient variability. And so we have 7 clinical partners today, but are quickly expanding and should have over 15 by the end of the year. These 165 unique patient tumors are not valuable just today to determine how predictive our platform is, but will continue to be powerful for us in years to come as we continue to build our data set and move into drug discovery. We've also, to date, raised just over $7 million. This capital has come locally from Utah investors as well as throughout the country, with lead investment from Caffeinated Capital, Coda Capital, and Khosla Ventures. We're excited to continue growing in Utah. And so in the next 2 years, we'll be continuing to develop those new clinical partnerships that I discussed, increasing our platform bandwidth and automation, and in-licensing our first asset as we're able to use the power of our platform to now move drugs forward into phase 2 and phase 3, and then move into discovery after that. We're excited to continue raising additional capital locally and internationally in the years to come. We're in the process of expanding our lab, what is now 7,000 square feet, into 36,000 square feet by the end of the year, staying here in the research park, and then increasing our employee count from 16 to 65 by the end of 2023. And so Utah has been incredibly generous to us and has allowed us to build a very exciting and cutting-edge company in which we're marrying data science and cancer biology to discover and develop the best drugs for every cancer. So thank you.